Hello everyone. This video tutorial presents an example of a metabolically activated chemical in the QSAR Toolbox. The abiotic and biotic transformation of chemicals is a very important step in the hazard assessment of chemicals, as in many cases it is not the chemical itself but its metabolites that are responsible for the toxic effect. Let's get started. The current example shows a case for evaluating the skin sensitization potential of a chemical taking into account its abiotic and biotic transformation. The target chemical is ethylpyrocatechol and is entered into the system by its CAS number 28930-20-3. There is no experimental skin sensitization data for the target chemical. In this case, we are interested to see if the chemical itself is capable to cause skin sensitization. For this purpose, we need to analyze the profiling results for the target chemical considering also its abiotic and biotic activation. To do that, first we define the target endpoint to indicate the relevant profilers and abiotic and biotic simulators for skin sensitization. The target endpoint is the EC3 endpoint associated with the local lymph node assay defined in this way. The profilers and simulators relevant for EC3 are highlighted in green. As can be seen, all relevant profilers are associated with protein binding, the molecular initiating event of skin sensitization. The relevant simulators in this case are the oxidation simulator and the skin metabolism simulator. Details about the simulator can be found in the About and in the documentation of the simulator. To analyze the profiling result of a parent chemical and its metabolites with respect to the defined target endpoint, we need to select all green profilers and simulators together. In this way, we will not only see the metabolites generated by the selected profilers, but also their profiling results according to the selected profilers. Click on Apply and wait until the result is displayed in the data matrix. In the data matrix we see one new level that appear, profiling with many sublevels associated with the type of the profilers and simulators which are applied. First two sublevels are associated with the profiling result of the parent chemical by the applied profilers. In a separate sublevel called metabolism/transformation, we can find the profiling result of the products generated by the selected simulators. For example, there are two metabolites generated by the oxidation simulator and one generated by the skin metabolism simulator. Double-clicking on the cell with the result opens a table with the 2D structures of the generated metabolites. The results of the applied profilers appear in the corresponding cell in the data matrix, where we see summarized information about the availability of protein binding alerts for the entire list of generated metabolites. The red color of the information in the cell indicates alerts. In this case, there is no protein binding alert for the parent chemical, but the generated oxidation and skin metabolites have protein binding alerts. To see more details about the metabolite profiling results, you need to double-click on a cell with a profiling result. Let us do this for the profiling result of the oxidation products by the profiler protein binding alerts for skin sensitization by Oasis. The first level shows the total number of oxidation products generated. The sublevels show the profiling result for each of the metabolites generated. The individual metabolites are represented by their SMILES notations. In this example, we see that both oxidation products bind to the protein due to the presence of the quinone type alert, which follows the Michael addition mechanism of interaction. More details can be shown if a SMILES of a metabolite is selected and we then ask for details. This action opens up the mechanistic rationale for interaction with proteins, which is supported by the literature. This new window is the profiling scheme browser, which displays various information about the applied profiling scheme and the explained alert. The top left section shows all the profiler categories, with the explained category of the quinone alert in bold. Below this section we see the metabolite highlighted in the corresponding alert. Here in the frame we see some sections with specific information. Let us go to the first section definition where we can see the structural requirements for the alert. This alert has 14 requirements that are logically ordered. No matter which of them is met, the structure will be profiled as quinone. In addition, the requirements that match the structure are indicated with a green checkmark, while the requirements that do not match are marked with a red cross. This example shows a case for evaluating the potential skin sensitization effect of a target chemical, taking into account its abiotic and biotic transformation. In the present case, the chemical is activated by oxidation and skin metabolism. Please note that the availability of alerts as a result of the profiling is not a prediction but only indication for possible effect. This information can be further used in the category definition phase, where analogs with the same mechanistic profile can be collected as a result of the same activation as the target chemical. You can find out how to do this in the tutorial category definition, considering metabolic activation of the target chemical. Congratulations! Now you are ready to learn more about the functionalities in Toolbox and how to use them. Contact QSAR Toolbox Help Desk for technical questions. If your question concerns policy or regulatory acceptance of Toolbox results within the EU legislations of ECHA's competence, please refer to the ECHA website directly.